Every living thing on this planet, from the smallest bacterium to the largest whale, shares something fundamental that connects them across billions of years of history. That connection is not merely philosophical or poetic, but written into the very molecules that make life possible. The story of biology is, in many ways, the story of how matter organized itself into increasingly complex patterns, capable of maintaining, replicating, and eventually understanding themselves. To grasp how this happened requires moving beyond simple categories and appreciating the deep chemical logic that underlies all biological phenomena. Life as we observe it today depends on carbon-based chemistry, and this dependency is not arbitrary. Carbon possesses a unique capacity among elements to form 4 stable bonds with other atoms, allowing it to create long chains, branch structures, and rings that can be modified in countless ways. This versatility means that carbon compounds can encode information, store energy, and create physical structures with a specificity that other elements cannot match. Silicon, often proposed as an alternative basis for life in speculative discussions, can also form 4 bonds. But silicon-silicon bonds are far less stable than carbon-carbon bonds in the presence of oxygen and water. The chemistry of life as it evolved on Earth is therefore not simply one option among many, but reflects real constraints imposed by physics and chemistry under the conditions that prevailed on our planet. Water plays an equally fundamental role in biological processes, and its properties are stranger than everyday familiarity might suggest. The water molecule is bent rather than linear, with the oxygen atom carrying a slight negative charge, and the hydrogen atoms carrying slight positive charges. This polarity allows water molecules to form hydrogen bonds with each other and with other polar molecules. These bonds are individually weak but collectively create the distinctive properties of liquid water, including its high heat capacity, its ability to dissolve a wide range of substances, and its unusual behavior upon freezing. Ice floats because water expands as it solidifies, a consequence of the hydrogen bond network creating a crystalline structure less dense than the liquid. This property has profound biological consequences because it means that bodies of water freeze from the top down, leaving liquid water beneath the ice where organisms can survive through winter. The molecules that carry genetic information, DNA and RNA, represent one of the most remarkable solutions to the problem of heredity that chemistry allows. Deoxyribonucleic acid consists of 2 strands wound around each other in a double helix, with each strand composed of a sugar phosphate backbone and attached nitrogenous bases. The bases come in 4 types: adenine, thymine, guanine, and cytosine. The key to DNA's function lies in the specificity of base pairing, where adenine pairs only with thymine and guanine pairs only with cytosine. This complementarity means that each strand contains all the information needed to reconstruct its partner, providing a mechanism for accurate copying during cell division. The discovery of this structure by James Watson and Francis Crick in 1953, building on X-ray diffraction data obtained by Rosalind Franklin and Maurice Wilkins, transformed biology into an information science. RNA differs from DNA in having ribose rather than deoxyribose as its sugar component, and in using uracil instead of thymine as one of its bases. These differences might seem minor but have significant functional consequences. RNA is less chemically stable than DNA, making it suitable for temporary rather than permanent information storage. RNA can also fold into complex 3-dimensional shapes that allow it to catalyze chemical reactions, a property that DNA lacks. This catalytic ability of RNA molecules discovered in the 1980s suggested a possible solution to a long-standing puzzle about the origin of life. If life requires both information storage and catalytic activity, and if modern cells use DNA for information and proteins for catalysis, which came first? The RNA world hypothesis proposes that early life forms used RNA for both functions, with DNA and proteins evolving later as specialized refinements. Proteins are the primary workhorses of cells, performing catalytic, structural, signaling, and transport functions. Each protein is a chain of amino acids folded into a specific 3-dimensional shape, and this shape determines the protein's function. 20 different amino acids are used in most proteins, each with a different side chain that gives it distinct chemical properties. Some side chains are hydrophobic and tend to cluster in the interior of folded proteins. Others are hydrophilic and remain on the exterior, interacting with the surrounding water. Still others can form chemical bonds with other amino acids, stabilizing the folded structure. The sequence of amino acids in a protein is determined by the sequence of nucleotides in the gene that encodes it, following a genetic code that is nearly universal across all life forms. The genetic code works through intermediate molecules called messenger RNAs, which are copies of gene sequences made in a process called transcription. These messenger RNAs are then read by ribosomes, complex molecular machines composed of both RNA and protein, in a process called translation. The ribosome reads the messenger RNA, 3 nucleotides at a time, with each triplet specifying a particular amino acid. Transfer RNA molecules deliver the appropriate amino acids to the ribosome, matching their anticodon sequences to the codons on the messenger RNA. This system is remarkably accurate, making fewer than 1 error per 10,000 amino acids incorporated, and remarkably fast, adding roughly 20 amino acids per second in bacterial cells. The universality of the genetic code across all known life forms is one of the strongest pieces of evidence for common descent. Bacteria, archaea, fungi, plants, animals, and protists all use essentially the same code, with only minor variations in a few lineages. This universality extends beyond the code itself to include many of the molecular machines involved in gene expression. The ribosome, for example, is remarkably similar in all organisms, with a core structure that has been conserved for billions of years. This conservation reflects both the functional importance of the ribosome and the difficulty of changing it, since any modification would need to remain compatible with the rest of the translation machinery. Cells, the basic units of life, come in 2 fundamentally different types. Prokaryotic cells, which include bacteria and archaea, lack membrane-bound internal compartments. Their DNA is located in a region called the nucleoid, rather than being enclosed in a nucleus. Eukaryotic cells, which include all animals, plants, fungi, and protists, have a true nucleus surrounded by a double membrane, as well as various other membrane-bound organelles. The distinction between prokaryotes and eukaryotes represents one of the deepest divisions in the living world. Although molecular phylogenetics has revealed that the relationship between these groups is more complex than once thought, the archaea, once grouped with bacteria as prokaryotes, are now recognized as a distinct domain of life, as different from bacteria at the molecular level as both are from eukaryotes. Indeed, some molecular features suggest that eukaryotes are more closely related to archaea than to bacteria, leading to hypotheses that eukaryotes evolved from within the archaeal domain through a merger with bacterial cells. This merger hypothesis is supported by the observation that eukaryotic cells contain mitochondria, organelles that carry their own small circular DNA and reproduce by division within the cell. Mitochondria are descended from free-living bacteria that entered into a symbiotic relationship with ancestral archaeal cells, a relationship that became so intimate over evolutionary time that the mitochondria can no longer survive outside their host cells. The evidence for the bacterial origin of mitochondria includes the circular nature of mitochondrial DNA, the similarity of mitochondrial ribosomes to bacterial ribosomes, and the double membrane that surrounds mitochondria, consistent with an engulfment event. The inner membrane of mitochondria is functionally similar to the plasma membrane of bacteria, while the outer membrane resembles the membrane of the engulfing cell. Similar evidence supports the bacterial origin of chloroplasts, the organelles that carry out photosynthesis in plants and algae. Chloroplasts are descended from cyanobacteria, and their acquisition by eukaryotic cells allowed the evolution of photosynthetic eukaryotes and ultimately the greening of the planet's surface. Photosynthesis itself is a process of remarkable complexity and efficiency. The basic reaction involves capturing light energy and using it to convert carbon dioxide and water into sugars and oxygen. This sounds simple but requires a series of precisely coordinated steps. Light is absorbed by pigment molecules, primarily chlorophylls, which are arranged in protein complexes called photosystems. The absorbed energy excites electrons to higher energy states, and these excited electrons are passed along chains of carrier molecules, ultimately driving the synthesis of ATP, the universal energy currency of cells, and NADPH, a carrier of reducing power, In a separate set of reactions, ATP and NADPH are used to fix carbon dioxide into organic molecules through a cycle of reactions discovered by Melvin Calvin in the 1950s. The oxygen produced by photosynthesis comes from water molecules that are split by a cluster of manganese atoms at the heart of photosystem 2. This water-splitting reaction is chemically difficult, because water molecules are very stable, and the evolution of this capability was a pivotal event in the history of life. Before oxygenic photosynthesis evolved, the atmosphere contained little or no free oxygen. The earliest photosynthetic organisms likely used hydrogen sulfide or other compounds as electron donors, producing sulfur rather than oxygen as a byproduct. Cyanobacteria, which evolved oxygenic photosynthesis perhaps 2.5 to 3 billion years ago, began releasing oxygen into the atmosphere, triggering one of the most dramatic environmental changes in Earth's history. The accumulation of atmospheric oxygen had profound consequences for life. Oxygen is chemically reactive and toxic to many organisms that had evolved in its absence. The rise of oxygen caused mass extinctions among anaerobic organisms, while creating new opportunities for those that could tolerate or exploit it. Aerobic respiration, the process by which cells extract energy from organic molecules using oxygen as the final electron acceptor, yields far more ATP per glucose molecule than anaerobic alternatives. This higher energy yield may have enabled the evolution of larger, more active and more complex organisms. The relationship between atmospheric oxygen and life has not been constant over geological time. Evidence from sedimentary rocks suggests that oxygen levels rose in stages, with the Great Oxidation event around 2.4 billion years ago, representing a major transition but not immediately achieving modern levels. Oxygen concentrations may have fluctuated considerably over the following billion years, a period sometimes called the boring billion, because it shows relatively little evidence of major evolutionary innovation. Only in the late Proterozoic, around 700 to 800 million years ago, did oxygen levels rise toward modern values, coinciding with the evolution of the first animals. The evolution of animals required not only sufficient oxygen, but also new ways of organizing cells into multicellular bodies. Multicellularity has evolved independently dozens of times in different lineages, but complex multicellularity with specialized tissues and organs is relatively rare. Animals, plants, fungi, and a few groups of algae have achieved this level of organization, but most multicellular lineages remain relatively simple. The transition to complex multicellularity requires mechanisms for cell adhesion, cell communication, and coordinated gene regulation that allow different cell types to develop and maintain their distinct identities while remaining parts of a unified organism. In animals, cell types are distinguished by the genes they express, which in turn are controlled by networks of regulatory proteins and non-coding RNAs. A skin cell and a nerve cell in the same body contain identical DNA, but differ dramatically in which genes are active. This differential gene expression is established during embryonic development and maintained throughout the life of the organism. The mechanisms that control gene expression include transcription factors that bind to specific DNA sequences, chromatin modifications that affect the accessibility of genes, and epigenetic marks that can be inherited through cell division without changes to the DNA sequence itself. Embryonic development in animals follows patterns that are surprisingly conserved across species. All bilaterally symmetrical animals, from insects to humans, share a common set of developmental genes that specify body axes and segment identities. The discovery of these conserved genes, particularly the Hox genes that control identity along the head-to-tail axis, revealed that animals that look completely different are using the same molecular toolkit assembled in different ways. A fly and a mouse both use Hox genes to pattern their body axes, even though the end results are dramatically different. This deep conservation of developmental mechanisms implies that the last common ancestor of these animals, living perhaps 600 million years ago, already possessed a sophisticated developmental program. The fossil record provides glimpses of early animal evolution, though the interpretation of these ancient remains is often contentious. The Ediacaran biota, found in rocks from roughly 575 to 540 million years ago, includes a variety of forms that have been variously interpreted as early animals as members of extinct kingdoms of life or as preservational artifacts. Some Ediacaran fossils, such as Dickinsonia, show features consistent with animal organisation, but their precise relationships remain debated. The Cambrian explosion, beginning around 540 million years ago, saw the rapid appearance of most modern animal phyla in the fossil record. This apparent burst of evolutionary innovation may reflect a genuine acceleration of evolution, an artifact of improved preservation conditions, or both. The Cambrian period produced many strange forms that have no obvious modern counterparts. The Burgess Shale of British Columbia and similar deposits around the world preserve animals with body plans that seem almost alien, such as Anomalocaris with its large size and grasping appendages, Opabinia with its 5 eyes and forward-pointing proboscis, and Hallucigenia with its spines and legs initially reconstructed upside down. These fossils demonstrate that the early history of animals was a time of evolutionary experimentation, with many body plans appearing that did not persist to the present. Whether this pattern reflects ecological opportunity in newly available niches, developmental flexibility before constraints became fixed, or simply the randomness of extinction is actively debated. The colonization of land by plants and animals transformed Earth's surface and created new evolutionary opportunities. Plants appear to have made the transition from water to land around 470 million years ago, probably from freshwater green algae. The challenges of terrestrial life include desiccation, the need to support body weight without water's buoyancy, and the difficulty of reproducing without an aqueous medium for sperm to swim through. Early land plants developed waxy cuticles to reduce water loss, stomata to regulate gas exchange, and vascular tissue to transport water and nutrients. The evolution of seeds allowed plants to disperse and reproduce without depending on free water, enabling the colonization of drier habitats. Animals followed plants onto land, with arthropods among the earliest colonizers. The oldest known terrestrial arthropods date to around 430 million years ago, though earlier colonization is possible. Vertebrates made the transition later, with the first tetrapods evolving from lobe-finned fishes around 375 million years ago. Fossils like Tiktaalik show intermediate forms with features of both fish and tetrapods, including fins with wrist-like structures and a neck that allowed the head to move independently of the body. These transitional forms illustrate how evolution proceeds through modification of existing structures rather than sudden creation of new ones. The conquest of land by vertebrates required numerous physiological adaptations. Gills, which extract oxygen from water, were replaced or supplemented by lungs, which extract oxygen from air. The circulatory system was modified to prevent mixing of oxygenated and deoxygenated blood, a process that reached its most complete form in the 4-chambered hearts of birds and mammals. Limbs evolved from fins, with the basic pattern of 1 bone, 2 bones, many bones, and digits appearing early and persisting in modified forms across all tetrapods. Skin became less permeable to reduce water loss, and kidneys became more efficient at conserving water and excreting nitrogenous wastes. Reproduction on land posed particular challenges that different lineages solved in different ways. Amphibians remain tied to water for reproduction, with most species producing eggs that must remain moist and larvae that develop in aquatic environments. Reptiles, birds, and mammals evolved the amniotic egg, a structure that contains the membranes and fluid necessary for embryonic development independent of external water. The amnion surrounds the embryo in a fluid-filled cavity, the chorion facilitates gas exchange, and the allantois collects metabolic wastes. This innovation allowed amniotes to reproduce in fully terrestrial environments and colonize habitats unavailable to amphibians. Mammals took the amniotic egg in a different direction, with most species retaining the embryo within the mother's body until birth. The placenta, a structure that facilitates exchange of nutrients, gases, and wastes between mother and fetus, allows prolonged internal development. This reproductive strategy permits greater parental investment in fewer offspring, a pattern associated with larger brains and more complex behaviours. Mammals also evolved endothermy, the ability to maintain a constant body temperature through metabolic heat production, which allows activity in a wider range of environmental conditions but requires higher food intake. The combination of endothermy, live birth, and extended parental care characterises most modern mammals, though monotremes like the platypus retain egg-laying as the ancestral condition. Birds represent a separate evolution of endothermy from within the dinosaurs, and indeed, under modern phylogenetic definitions, birds are dinosaurs. The evolution of flight required extensive modifications to the skeletal and muscular systems, including hollow bones to reduce weight, a keeled sternum for flight muscle attachment, and fusion of skeletal elements to provide rigidity. Feathers, which likely evolved originally for insulation or display, were co-opted for aerodynamic function. The earliest known bird, Archaeopteryx, lived about 150 million years ago and possessed a mixture of dinosaurian features like teeth and a bony tail with bird-like features like feathers and a wishbone. Subsequent discoveries of feathered dinosaurs in China have blurred the line between birds and their relatives, showing that many dinosaurs were feathered and that flight may have evolved multiple times. The diversity of life today reflects billions of years of evolutionary history, during which lineages have diverged, adapted, and sometimes gone extinct. Extinction is a normal part of evolutionary history, with the vast majority of species that have ever lived now extinct. Background extinction rates, the normal turnover of species over geological time, are punctuated by mass extinctions, events in which a significant fraction of species disappear in a geologically short period. 5 major mass extinctions are recognised in the past 540 million years, the most severe being the end-Permian extinction about 252 million years ago, which eliminated perhaps 90% of marine species and 70% of terrestrial vertebrate species. The causes of mass extinctions vary and are often debated. The end Cretaceous extinction, which eliminated non-avian dinosaurs along with many other groups about 66 million years ago, is now generally attributed to an asteroid impact, with supporting evidence including a global layer of iridium-enriched clay at the appropriate geological boundary and the Chicxulub crater in Mexico, dating to the same time. Other mass extinctions have been linked to volcanic eruptions, climate change, changes in ocean chemistry, or combinations of factors. The end-Permian extinction coincided with massive volcanic eruptions in Siberia that released enormous quantities of carbon dioxide and other gases, triggering global warming, ocean acidification, and oxygen depletion. Understanding past extinctions is relevant to understanding present-day biodiversity loss, though the mechanisms differ. Current extinction rates are estimated to be 100 to 1,000 times higher than background rates, driven primarily by habitat destruction, overexploitation, introduced species, pollution, and climate change. These are fundamentally different from the causes of past mass extinctions, representing the impact of a single species rather than external physical events. Whether current biodiversity loss will ultimately reach mass extinction proportions depends on human actions over the coming decades and centuries. The study of biodiversity has been transformed by molecular techniques that allowed direct comparison of DNA sequences across species. Before molecular phylogenetics, classification relied on observable physical features, which could be misleading when similar structures evolved independently in unrelated lineages. Molecular data provide a more direct window into evolutionary relationships, revealing unexpected connections and overturning long-held assumptions. The close relationship between hippopotamuses and whales, for example, was established by molecular evidence before fossil intermediates were discovered. Similarly, molecular data confirmed the dinosaurian origin of birds and revealed that fungi are more closely related to animals than to plants. The tree of life, as currently understood, consists of 3 domains: bacteria, archaea, and eukarya. Within eukarya, the relationships among major groups have been substantially revised by molecular studies. Traditional classification recognized plants, animals, fungi, and protists as major kingdoms, but protists are now understood to represent multiple independent lineages rather than a coherent group. The distribution of mitochondria and plastids across eukaryotic lineages tells a story of symbiosis and lateral gene transfer that complicates simple branching diagrams. Some lineages have acquired plastids multiple times through secondary or tertiary endosymbiosis, engulfing already photosynthetic eukaryotes rather than free-living cyanobacteria. The mechanisms of evolution have been refined since Darwin's original formulation. Natural selection remains the primary mechanism for adaptation, but other processes also contribute to evolutionary change. Genetic drift, the random fluctuation of allele frequencies in finite populations, can lead to evolutionary change that is not adaptive. In small populations, drift can be powerful enough to overcome selection, leading to the fixation of mildly deleterious alleles. This has implications for conservation biology, where small population sizes in endangered species may reduce their adaptive potential, even if immediate extinction is avoided. Mutation provides the raw material for evolution, introducing new genetic variation on which selection and drift can act. Mutation rates vary among organisms and among different regions of the genome. DNA repair mechanisms keep mutation rates low enough that genomes remain functional across generations, but high enough to provide variation for evolution. Some mutations are neutral, having no effect on fitness. Others are deleterious and are removed by natural selection. Rarely mutations are beneficial, improving the organism's ability to survive and reproduce in its current environment. The rate at which beneficial mutations arise and spread through populations limits how quickly evolution can proceed. Recombination shuffles existing genetic variation, creating new combinations of alleles that can be tested by selection. Sexual reproduction, which involves recombination, is nearly universal among eukaryotes despite the twofold cost of producing males who do not themselves bear offspring. The persistence of sex is explained by several hypotheses, including the advantage of creating genetically variable offspring that can resist parasites and pathogens, the ability to purge deleterious mutations from the genome, and the faster spread of beneficial mutations when recombination can bring them together on the same chromosome. Gene flow, the movement of alleles between populations, can homogenize populations or introduce new variation. In species with high dispersal, gene flow may prevent local adaptation by swamping locally beneficial alleles, with immigrants carrying alleles adapted to other environments. In species with limited dispersal, populations may diverge genetically and eventually become reproductively isolated, forming new species. The process of speciation, by which one species becomes two, can occur in various ways. Allopatric speciation, in which populations are geographically separated, is probably the most common mechanism, but speciation can also occur without complete geographic isolation through ecological or sexual selection. The diversity of species on Earth is unevenly distributed, with tropical regions generally containing more species than temperate or polar regions. This latitudinal gradient in diversity has been recognized for centuries, but remains incompletely explained. Hypotheses include greater energy input and productivity in the tropics, greater environmental stability allowing more specialization, older tropical biomes accumulating more species over time, and geometric effects related to the larger tropical area. These explanations are not mutually exclusive, and the gradient likely results from multiple interacting factors. Within any community, species interact through competition, predation, parasitism, mutualism, and other relationships that influence their distributions and abundances. Ecological theory attempts to understand how these interactions structure communities and how many species can coexist in a given habitat. The competitive exclusion principle suggests that two species with identical ecological niches cannot coexist indefinitely, as one will inevitably outcompete the other. Observed coexistence implies niche differentiation, with species differing in their resource use, habitat preferences, or timing of activity. Alternatively, coexistence can be maintained by mechanisms that prevent any single species from dominating, such as predation on the competitive dominant or environmental fluctuations that favour different species at different times. Food webs describe the feeding relationships within communities, linking producers, consumers, and decomposers. Energy flows through these webs, entering as sunlight captured by photosynthesis and dissipating at each trophic level as organisms use energy for their own metabolism. The efficiency of energy transfer between levels, typically around 10%, limits the number of trophic levels that can be sustained and explains why top predators are rare. Nutrients, in contrast to energy, cycle through ecosystems, being taken up by organisms, incorporated into biomass, released by decomposition, and taken up again. Understanding these cycles is essential for managing ecosystems, as human activities often disrupt nutrient flows through fertilizer application, waste disposal, or changes in land use. Microorganisms play crucial roles in nutrient cycling that were long underappreciated because bacteria and archaea are invisible to the naked eye and difficult to culture in laboratories. Nitrogen-fixing bacteria convert atmospheric nitrogen gas into ammonia, a form that plants can use. Nitrifying bacteria oxidize ammonia to nitrite and nitrate, while denitrifying bacteria return nitrogen to the atmosphere by reducing nitrate to nitrogen gas. These microbial transformations control the availability of nitrogen, often the limiting nutrient for plant growth. Similarly, bacteria and archaea mediate the cycling of sulfur, phosphorus, iron, and many other elements, connecting the living world to the geochemistry of the planet. The human microbiome, the community of microorganisms living in and on our bodies, has attracted intense research interest in recent years. The number of bacterial cells in a typical human body roughly equals the number of human cells, though human cells are much larger and constitute most of the body mass. The gut microbiome influences digestion, immunity, metabolism, and even brain function, through pathways that are still being elucidated. Different individuals harbour different microbial communities shaped by genetics, diet, medication history, and early life experiences. Disruption of the microbiome through antibiotic use or dietary changes has been implicated in various health conditions, though establishing causation is challenging. The immune system represents one of the most complex and sophisticated biological systems, capable of distinguishing self from non-self and mounting targeted responses against pathogens while avoiding autoimmune attack on the body's own tissues. Vertebrate adaptive immunity relies on lymphocytes bearing receptors of staggering diversity, generated through random rearrangement of gene segments. Each lymphocyte carries receptors of a single specificity, and when it encounters a matching antigen, it proliferates to generate many cells capable of recognizing that antigen. This clonal selection mechanism allows the immune system to respond to virtually any pathogen, including novel ones never previously encountered, while maintaining memory of past infections that provides lasting protection. Pathogens, in turn, have evolved numerous strategies to evade or suppress immune responses. Viruses may mutate rapidly, preventing the immune system from recognizing them based on prior exposure. Bacteria may coat themselves with capsules that hide surface molecules from immune detection. Parasites may suppress host immune responses or sequester themselves in locations the immune system cannot easily reach. The ongoing evolutionary arms race between pathogens and immune systems has shaped the genetics of both, with signatures of this conflict visible in patterns of variation at immune-related genes. Viruses occupy an unusual position in biology, possessing some but not all of the features typically associated with life. Viruses contain genetic information and can evolve, but they cannot replicate independently and must hijack the cellular machinery of their hosts. Whether viruses should be considered alive or not is largely a semantic question, but their importance to biology is undeniable. Viruses infect all forms of cellular life and may outnumber cells in many environments. Viral infections have shaped the evolution of their hosts in profound ways, and viral sequences have been incorporated into host genomes throughout evolutionary history. Endogenous retroviruses, the remnants of ancient viral infections, make up about 8% of the human genome. The human genome, fully sequenced in 2003, contains roughly 20,000 protein coding genes, fewer than initially expected for an organism of our complexity. Much of the genome consists of non-coding sequences, including regulatory elements, introns within genes, transposable element remnants, and sequences of unknown function. The ENCODE project and related efforts have suggested that much of this non-coding DNA is transcribed and may have functional roles, though the extent of true function versus biological noise remains debated. Comparative genomics, comparing genomes across species, reveals which sequences are conserved and therefore likely functional, as well as which sequences vary and may contribute to phenotypic differences. Genetic variation within humans is small compared to many other species, reflecting our relatively recent origin and history of population bottlenecks. All humans share over 99% of their DNA sequence, with most variation consisting of single nucleotide differences. Despite this overall similarity, genetic variation influences many traits, from disease susceptibility to physical appearance to drug response. Genome-wide association studies have identified thousands of genetic variants associated with various traits and diseases, though most individual variants have small effects and explain only a fraction of heritability. Complex traits like height, intelligence, or susceptibility to common diseases are influenced by many genes and environmental factors, making prediction from genetics alone challenging. The development of CRISPR-Cas gene editing technology has revolutionized biology's ability to modify genomes with precision. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, is a bacterial immune system that was co-opted as a laboratory tool. By programming the CRISPR system with a guide RNA matching any target sequence, researchers can cut DNA at specific locations and introduce desired changes. This technology has accelerated research across biology and raised both hopes and concerns about applications in medicine, agriculture, and beyond. The ethical implications of gene editing in humans, particularly in the germline where changes would be inherited by future generations, are actively debated by scientists, policymakers, and the public. Synthetic biology takes genetic engineering further, aiming to design and construct new biological parts, devices, and systems. This field draws on engineering principles, treating biological components as modular elements that can be combined in novel ways. Applications include microorganisms engineered to produce fuels, chemicals, or pharmaceuticals, biosensors for detecting environmental contaminants, and potential future technologies like programmable living materials. The long-term trajectory of synthetic biology is uncertain, but the field represents a qualitatively new relationship between humans and the biological world, moving from modification of existing organisms to design of new ones. Understanding biology at the molecular level requires understanding how molecules interact within cells. Proteins do not function in isolation, but as parts of networks, with each protein interacting with numerous partners in ways that can be activating or inhibiting, transient or stable. Signal transduction pathways transmit information from cell surface receptors to target genes, often involving cascades of protein modifications that amplify and diversify the signal. These pathways are not simple linear chains, but interconnected networks with feedback loops, crosstalk, and redundancy that make them robust to perturbation, but difficult to fully characterize. Cancer represents a disease of cellular control gone wrong, with cells dividing uncontrollably, evading normal growth limits, and sometimes spreading to distant sites. Cancer arises through accumulation of mutations in genes that regulate cell growth, division, and death. Oncogenes, when mutated or overexpressed, promote cell division, while tumor suppressor genes normally restrain division and must be inactivated for cancer to develop. The multi-step nature of cancer development, requiring multiple mutations, explains why cancer typically arises late in life as mutations accumulate, and why inherited mutations in tumor suppressor genes greatly increase cancer risk by providing a head start. The immune system normally recognizes and destroys abnormal cells, including early cancers, but advanced tumors have evolved mechanisms to evade immune surveillance. Cancer immunotherapy aims to overcome this evasion by boosting the immune response against tumors. Checkpoint inhibitors, which release the brakes that tumors apply to immune cells, have produced remarkable responses in some patients with cancers previously considered untreatable. Other approaches include engineering patients' own immune cells to recognize tumor-specific antigens and vaccinations that prime the immune system against cancer. These therapies represent a fundamentally different approach to cancer treatment, targeting the host's response rather than the tumor directly. Developmental biology addresses how a single fertilized egg gives rise to an organism with trillions of cells, organized into complex structures. The early embryo is remarkably flexible, with cells able to adopt different fates depending on their position and the signals they receive. This plasticity diminishes as development proceeds, and cells become committed to particular lineages. The mechanisms that control these decisions involve gradients of signaling molecules, cascades of transcription factor activation, and chromatin modifications that lock in gene expression patterns. The same basic mechanisms operate across the animal kingdom, modified in different lineages to produce the diversity of body plans observed. Stem cells retain developmental plasticity even in adult organisms, serving as reservoirs for tissue renewal and repair. Hematopoietic stem cells in bone marrow produce all blood cell types throughout life. Intestinal stem cells replace the gut lining every few days. Neural stem cells in certain brain regions continue to generate new neurons in adults, though the extent and importance of adult neurogenesis remain debated. The discovery that mature cells can be reprogrammed to a pluripotent state by introducing just 4 transcription factors by Shinya Yamanaka in 2006 opened new possibilities for regenerative medicine and disease modelling. Neuroscience seeks to understand how the brain, an organ of roughly 86 billion neurons connected by trillions of synapses, generates perception, thought, and behaviour. Neurons communicate through electrochemical signals, with action potentials travelling along axons and neurotransmitters, crossing synaptic clefts to excite or inhibit target cells. Neural circuits, composed of interconnected neurons with specific patterns of connectivity, perform computations that underlie sensory processing, motor control, and cognition. Understanding how these circuits work requires integrating information across scales, from molecular properties of ion channels to whole brain activity patterns measured by neuroimaging. The brain is not a uniform structure, but consists of distinct regions with different architectures and functions. The cerebral cortex, the outermost layer, is responsible for perception, thought, and voluntary action in mammals. Different cortical areas specialize in different functions, with primary sensory areas receiving input from eyes, ears, and touch receptors, and motor areas controlling muscles. Association areas integrate information across modalities and support higher cognitive functions. Subcortical structures like the hippocampus are essential for memory formation, while the amygdala processes emotional information and the basal ganglia coordinate movement and reward learning. Learning and memory involve changes in the strength of synaptic connections, allowing experience to modify neural circuits. Long-term potentiation, an increase in synaptic strength following repeated activation, was first described in the hippocampus and is believed to underlie some forms of memory. At the molecular level, long-term potentiation involves changes in the number and properties of receptor proteins at synapses, stabilized by protein synthesis and structural changes in dendritic spines. Different types of memory involve different brain regions and molecular mechanisms, with procedural memories for skills depending on different circuits than declarative memories for facts and events. Sleep, though universal among animals with nervous systems, remains incompletely understood in terms of its function. Sleep deprivation impairs cognition, immunity, and many other functions, indicating that sleep is essential, but the specific processes that require sleep are debated. Hypotheses include memory consolidation, synaptic homeostasis, metabolic waste clearance, and immune function. Different stages of sleep have different characteristics and likely different functions, with REM sleep particularly associated with dreaming and with memory consolidation for certain types of learning. The molecular mechanisms that regulate sleep and wakefulness involve neurotransmitter systems and circadian clock genes that have been identified through studies in model organisms. Circadian rhythms, the roughly 24-hour cycles in physiology and behavior, are controlled by molecular clocks within cells. In mammals, the master clock resides in a small group of neurons in the hypothalamus, which synchronizes peripheral clocks throughout the body. The molecular clock consists of transcription factors that activate their own repressors, creating a negative feedback loop with a period of approximately 24 hours. This timing mechanism is remarkably conserved across species, with homologous clock genes found in organisms from fungi to fruit flies to humans. Disruption of circadian rhythms through shift work, jet lag, or genetic mutation has widespread effects on health and disease. Behavior, the observable output of nervous system function, has evolved under natural selection just as morphology has. Behavioral ecology examines how behavior contributes to fitness, using evolutionary theory to predict optimal strategies in foraging, mate choice, parental care, and social interaction. Animals often behave in ways that appear altruistic, benefiting others at cost to themselves, which seems to contradict natural selection's focus on individual fitness. Kin selection explains altruism among relatives, whose shared genes mean that helping relatives can spread copies of the altruist's genes. Reciprocal altruism explains cooperation among non-relatives when individuals interact repeatedly and can punish cheaters. Social insects like ants, bees, and termites display extreme cooperation, with sterile workers forgoing reproduction to assist their colony's queen. The evolution of eusociality, this level of cooperative breeding, is explained partly by the unusual genetic systems of some social insects. In hymenopteran insects like ants and bees, females are more closely related to their sisters than to their own offspring due to their haplodiploid genetics, potentially favouring helping over independent reproduction. However, eusociality has also evolved in termites, which are diploid, and in naked mole rats, suggesting that high relatedness is not the only factor. Ecological constraints that make independent reproduction difficult may also be important. Communication among animals takes many forms, from the dances of honeybees that convey the location of food sources to the complex vocalizations of songbirds and whales. Human language appears qualitatively different from other animal communication systems in its generativity, its ability to create unlimited novel utterances from a finite set of elements. The evolution of language is difficult to study because it leaves no direct fossil record, though anatomical features like the descended larynx and the articulation of the hyoid bone provide indirect evidence. Comparative studies of communication in other species, particularly our closest relatives, the great apes, inform understanding of what cognitive capabilities might have preceded human language. Cognition, the mental processes that underlie behavior, has become accessible to experimental study in animals through carefully designed behavioral tests. Studies have demonstrated impressive cognitive abilities in many species, including tool use and manufacture in chimpanzees and crows, mental time travel in scrub jays that hide food for future use, numerical abilities in various primates and birds, and theory of mind, the ability to understand others' mental states, in apes and possibly other animals. These findings have blurred the line between human and animal cognition, suggesting that many abilities once considered uniquely human exist in some form in other species and likely evolved gradually. Human evolution has become increasingly well documented through fossil discoveries, genetic analyses, and comparisons with other primates. The lineage leading to humans diverged from that leading to chimpanzees and bonobos approximately 6 to 7 million years ago. Early hominins like Ardipithecus and Australopithecus were bipedal, but retained small brains and ape-like faces. The genus Homo appeared around 2.5 million years ago, with subsequent species showing increasing brain size, reduced faces and teeth, and increasingly sophisticated tools. Modern humans, Homo sapiens, evolved in Africa around 300,000 years ago and dispersed to other continents within the last 100,000 years. The relationship between modern humans and other recent human species is being rewritten by ancient DNA studies. Neanderthals, who inhabited Europe and Western Asia until around 40,000 years ago, were long considered entirely separate from our lineage. However, genomic analysis of Neanderthal bones revealed that most people of non-African descent carry 1 to 4% Neanderthal DNA, indicating interbreeding when modern humans expanded out of Africa. Similarly, the Denisovans, known primarily from DNA rather than fossils, contributed genes to populations in Oceania and mainland Asia. These genetic contributions persist because some Neanderthal and Denisovan genes provided advantages to modern humans, including genes affecting immunity and adaptation to high altitude. Human cultural evolution operates by mechanisms distinct from genetic evolution, but interacting with it. Cultural practices can change within generations, spreading through learning and imitation rather than genetic inheritance. This has allowed humans to adapt to environments far beyond the range of our biological tolerances, through technologies like clothing, shelter, and fire. Cultural evolution can also drive genetic evolution, as in the case of lactase persistence. the ability to digest milk in adulthood, which evolved in populations with long histories of pastoralism. Gene culture coevolution is a distinctly human phenomenon, though other animals show cultural transmission of behaviours. Agriculture, which began independently in several regions around 10 to 12,000 years ago, transformed human societies and their relationship to the environment. The cultivation of plants and domestication of animals allowed permanent settlements, population growth, and eventually complex societies with division of labour, social hierarchies, and specialised institutions. Agricultural populations faced new disease pressures as infectious diseases spread more easily in dense settlements and jumped to humans from domesticated animals. The dietary shift toward grains and the physical labour of farming also affected human health in ways still detectable in skeletal remains. The biological effects of agriculture and subsequent technological changes continue to shape modern humans. Population sizes have increased dramatically, reducing the effects of genetic drift and increasing the efficiency of selection against deleterious mutations. At the same time, relaxation of natural selection through medical care, sanitation, and food security may allow harmful mutations to accumulate. Changes in diet, physical activity, and exposure to infectious diseases have created mismatches between our evolved biology and modern environments, contributing to chronic diseases like obesity, diabetes, and heart disease. Understanding these evolutionary mismatches provides insights for public health and individual health decisions. Ecology at larger scales examines how ecosystems function and respond to change. Ecosystems receive energy from the sun, cycle nutrients through biological and geological reservoirs, and maintain conditions that support their component species. These functions are performed by the organisms present, but not all species contribute equally. Keystone species have effects disproportionate to their abundance, such as predators that prevent competitive exclusion or ecosystem engineers that create habitat for others. The loss of keystone species can trigger cascading changes throughout the ecosystem, as demonstrated by the consequences of removing wolves from Yellowstone or sea otters from kelp forests. Ecosystem services, the benefits that humans derive from functioning ecosystems, include provisioning services like food and clean water, regulating services like climate moderation and flood control, and cultural services like recreation and aesthetic value. Valuing these services economically is challenging but important for incorporating them into decision-making. Many ecosystem services have no market price, leading to their undervaluation and overexploitation. The degradation of ecosystem services through habitat destruction, pollution, and climate change imposes costs that are often borne by different people or at different times than those receiving the benefits. Biogeochemical cycles connect the living and non-living components of the Earth system. The carbon cycle involves exchange of carbon among the atmosphere, oceans, land surface, and deep geological reservoirs. Before human influence, these exchanges were roughly in balance, but fossil fuel combustion has transferred carbon from geological reservoirs to the atmosphere faster than natural processes remove it. Atmospheric carbon dioxide concentrations have risen from about 280 parts per million before industrialization to over 420 parts per million today, higher than at any time in millions of years. This change is driving global climate change through the enhanced greenhouse effect, as carbon dioxide absorbs infrared radiation and warms the atmosphere. Climate change affects biology in numerous ways. Species distributions are shifting toward poles and to higher elevations as temperatures rise. The timing of seasonal events like flowering and migration is advancing, sometimes creating mismatches between interacting species. Ocean acidification, caused by absorption of atmospheric carbon dioxide, threatens marine organisms that build shells or skeletons of calcium carbonate. Coral reef ecosystems, among the most biodiverse on Earth, face particular risks from the combination of warming and acidification. The rates of these changes exceed those of most past climate shifts, challenging the ability of species to adapt or migrate. Conservation biology applies scientific understanding to the preservation of biodiversity. The field integrates ecology, evolution, genetics, and other disciplines to inform management decisions. Conservation priorities often focus on hotspots, regions with high concentrations of endemic species facing significant threats. Protected areas remain important tools for conservation, but their effectiveness depends on size, connectivity, and management. Landscape-level approaches that maintain corridors between protected areas and compatible land uses in surrounding regions may be necessary for long-term viability of populations. The biology of plants underlies primary production and shapes terrestrial ecosystems. Plant physiological processes, including photosynthesis, water transport, and nutrient acquisition, determine productivity and growth patterns. The vascular system of plants, consisting of xylem that transports water upward and phloem that distributes sugars, allows trees to grow over 100 meters tall, though physical limits on water transport may ultimately constrain maximum height. Root systems explore soil for water and nutrients, often in partnership with mycorrhizal fungi that extend the effective reach of roots in exchange for carbon from the plant. Plant defenses against herbivores and pathogens illustrate evolutionary arms races. Chemical defenses, including toxins, digestibility reducers, and feeding deterrents, have evolved repeatedly across plant lineages. Many of these compounds have found uses in human medicine and industry, from aspirin derived from willow bark to Taxol from Pacific yew to rubber from rubber trees. Some plants mount induced defenses that are produced only after herbivore damage, conserving resources when enemies are absent. Others emit volatile signals that attract predators or parasitoids of their herbivores, recruiting enemies of their enemies. Plant reproduction shows remarkable diversity, from the wind-pollinated grasses that include our major grain crops to the elaborate flowers that attract specific animal pollinators. The evolution of flowers, about 140 million years ago, was followed by rapid diversification of flowering plants into the dominant land plant group. The intimate relationship between flowers and their pollinators has driven coevolution, with flower shapes, colours, and scents adapted to particular pollinators, and pollinators adapted to exploit particular flowers. Some of these relationships are highly specific, with plants pollinated by only one or a few species, while others are generalized, with many different animals visiting the same flowers. Seed dispersal similarly involves diverse mechanisms and ecological interactions. Wind dispersal favors light seeds with wings or plumes, while animal dispersal often involves fleshy fruits that reward the disperser with food. Some plants eject their seeds explosively, while others float on water or attach to passing animals. The distance that seeds travel affects population dynamics and genetic structure, with long-distance dispersal allowing colonization of new sites, but potentially breaking up locally adapted genotypes. Climate change is creating challenges for dispersal-limited species that cannot track suitable conditions as they shift geographically. Marine biology encompasses the vast majority of Earth's biosphere by volume, though most biological diversity by species numbers is on land. The oceans contain representatives of most major animal phyla, many of which have no terrestrial representatives. Ocean ecosystems range from productive coastal zones enriched by nutrient upwelling to oligotrophic open ocean gyres where productivity is limited by nutrient scarcity. The deep sea, below the euphotic zone where light can support photosynthesis, depends on organic matter sinking from surface waters or, in hydrothermal vent communities, on chemosynthesis by bacteria that oxidize hydrogen sulfide. Freshwater habitats, though covering only a tiny fraction of Earth's surface, harbour a disproportionate share of biodiversity, particularly among fish. Freshwater ecosystems are also among the most threatened, facing pressures from water extraction, pollution, dam construction, invasive species, and climate change. Rivers connect terrestrial and marine ecosystems, transporting water, sediments, nutrients, and organisms. Dams interrupt these connections, fragmenting habitats for migratory fish, and altering downstream flows and sediment dynamics. The ecology of freshwater systems is tightly linked to the watersheds that feed them, making management challenging when watershed lands are under different ownership or jurisdictions. Microbial ecology has been transformed by molecular methods that allow identification and study of organisms that cannot be cultured in laboratories. Traditional microbiology required growing organisms on artificial media, but most microbes in nature do not grow under standard laboratory conditions. metagenomic approaches sequence all the DNA in an environmental sample, revealing the diversity and metabolic potential of entire microbial communities. These studies have revealed that microbial diversity far exceeds what was previously appreciated, and that microbes play even more varied ecological roles than assumed. Extremophiles, organisms that thrive in conditions once considered incompatible with life, have expanded understanding of life's limits. Thermophiles grow at temperatures above 80 degrees Celsius, with some archaea surviving above 120 degrees near hydrothermal vents. Halophils tolerate salt concentrations that would desiccate most cells. Acidophils and alkalophils live at pH extremes that would denature most proteins. Pressure-tolerant organisms inhabit the deepest ocean trenches. These discoveries have implications for astrobiology, the search for life beyond Earth, suggesting that life might persist in environments once considered too harsh. Astrobiology looks for signatures of life on other planets and moons, guided by understanding of life on Earth. The requirements for life, as we know it, include liquid water, organic molecules, and energy sources. Within our solar system, Mars, the subsurface oceans of Europa and Enceladus, and even the clouds of Venus have been proposed as possible habitats for life. Detection of biosignatures, chemical or physical traces that indicate biological activity, is technically challenging and requires ruling out non-biological explanations. The discovery of life elsewhere would have profound implications for biology, demonstrating that life can originate independently and potentially revealing alternative biochemistries. The origin of life on Earth remains one of biology's greatest unsolved problems. The transition from non-living chemistry to living cells involved multiple steps, including the synthesis of organic molecules, the formation of polymers capable of storing information and catalyzing reactions, the encapsulation of these molecules within membranes, and the coupling of metabolism to replication. Different research programs focus on different aspects of this problem, from prebiotic chemistry that asks what molecules could have formed on early Earth, to protocell research that asks how simple compartments could have acquired the properties of cells. The Miller-Urey experiment of 1953 demonstrated that amino acids and other organic molecules could form under conditions simulating early Earth's atmosphere, though current understanding suggests Earth's early atmosphere differed from Miller and Urey's assumptions. Subsequent research has identified multiple pathways for synthesizing organic molecules, including delivery by meteorites and synthesis at hydrothermal vents. The RNA world hypothesis suggests that RNA preceded both DNA and proteins as the informational and catalytic molecule, but how RNA or its precursors originated remains unclear. The difficulty of these problems reflects the vast gap between the simplest known cells, which are still enormously complex, and any plausible prebiotic chemistry. Understanding biology requires appreciating that living systems operate far from thermodynamic equilibrium, maintained by constant input of energy. Cells actively transport molecules against concentration gradients, synthesize complex molecules from simpler precursors, and maintain structural organization despite the tendency toward disorder. This requires free energy, obtained from sunlight in photosynthetic organisms and from oxidation of organic molecules in most others. The coupling of energy-releasing reactions to energy-requiring processes is achieved through common intermediates, particularly ATP, whose hydrolysis releases energy that can drive otherwise unfavorable reactions. The second law of thermodynamics, which states that entropy tends to increase in isolated systems, is sometimes misconstrued as prohibiting the order observed in living systems. This reflects a misunderstanding. Organisms are not isolated systems. They continuously exchange matter and energy with their environments. The local decrease in entropy within a cell is more than compensated by the increase in entropy in its surroundings, satisfying the second law. Life does not violate thermodynamics but exploits it, using energy flow to create and maintain order. Information is a concept that has become central to biology, particularly since the molecular revolution. Genes carry information specifying proteins. Signaling pathways transmit information from cell surface to nucleus. Nervous systems process information to generate behavior. But what exactly information means in these biological contexts is not always clear. Information theory, developed by Claude Shannon for telecommunications, provides precise definitions applicable to some biological questions such as the information content of a DNA sequence. Whether these formal definitions capture everything biologists mean by information remains debated. Evolution generates biological information through the interaction of variation and selection. Random mutations introduce variation, most of which is neutral or harmful. Selection retains variants that contribute to survival and reproduction, gradually accumulating information about the environment in the gene sequences of populations. This process requires no foresight or intention, operating through the differential survival and reproduction of organisms with different heritable traits. The information in a genome reflects past selection pressures, not current or future environments, which is why organisms can be maladapted when environments change. The molecular clock hypothesis, that mutations accumulate at approximately constant rates over evolutionary time, provides a tool for dating evolutionary divergences. If the rate of molecular evolution is known, the number of differences between sequences can be converted to time since divergence. In practice, rates vary among genes, lineages, and time periods, requiring calibration with fossil evidence and careful statistical treatment. Despite these complications, molecular clocks have contributed importantly to understanding evolutionary timescales, often revealing divergences older than the fossil record indicated. Comparative biology reveals patterns in how traits are distributed across species, informing understanding of evolutionary history and adaptive function. Phylogenetic methods account for the non-independence of species due to shared ancestry, avoiding the statistical pitfalls of treating related species as independent data points. These methods have confirmed long-suspected correlations, like that between brain size and social group size in primates, while revealing unexpected patterns and refuting some prior claims. The integration of phylogenetic thinking into all areas of biology has been one of the major developments of recent decades. Systems biology takes a holistic approach to understanding biological systems, using mathematical models and high-throughput data to characterize the behavior of entire networks rather than individual components. This approach recognizes that properties of living systems emerge from the interactions among their parts and cannot always be predicted from studying parts in isolation. Computational models of metabolic networks, signaling pathways, and gene regulatory circuits can simulate cellular responses to perturbations and generate hypotheses for experimental testing. The goal is predictive understanding of complex biological systems, though current models remain limited in their predictive power for most systems. Quantitative methods have become increasingly important across biology. Statistical analysis distinguishes signal from noise in experimental data and observational studies. Mathematical models formalize verbal hypotheses and allow derivation of predictions that can be tested. Computation enables analysis of datasets too large for human inspection and simulation of systems too complex for analytical solutions. The integration of quantitative approaches with traditional biological methods has expanded what questions can be addressed and what answers can be supported with evidence. The funding and organization of biological research shape what questions are asked and how resources are allocated. Most basic biological research is funded by governments through agencies like the National Institutes of Health and National Science Foundation in the United States, analogous bodies in other countries, and international organizations. Funding priorities reflect societal concerns, scientific opportunities, and political processes that do not always align with scientific importance. Large collaborative projects like genome sequencing initiatives require coordination across many laboratories and institutions. Small investigator-initiated projects remain important for exploratory research and novel ideas. Ethical considerations pervade biological research and application. Animal welfare concerns shape how research using animals is conducted and regulated. Human subjects research follows principles developed in response to historical abuses, including voluntary informed consent and independent review of research protocols. Dual-use research, which could be misused to cause harm, raises questions about what research should be done and how results should be shared. Intellectual property rights in biological discoveries affect incentives for research and access to resulting products. These ethical considerations are not peripheral to biology but central to how it is practiced and applied. Biological education prepares future researchers and professionals while contributing to public understanding. Training in biology has evolved as the field has changed, with increasing emphasis on quantitative skills, interdisciplinary connections, and authentic research experiences. Public understanding of biology influences personal health decisions, policy choices, and cultural attitudes toward nature and technology. Misconceptions about evolution, genetics, and other topics remain common, with implications for issues ranging from vaccine acceptance to environmental policy. Effective science communication, including through popular science media, contributes to an informed public discourse. The history of biology reveals how current understanding was achieved and helps avoid reinventing discarded ideas. Scientific progress has not been linear, with periods of rapid advance interspersed with stagnation, wrong turns occasionally requiring correction, and ideas once rejected sometimes proving right. Understanding this history includes recognizing contributions from many cultures and the social context that shaped scientific development. The history of biology is not merely antiquarian interest, but informs appreciation of current science and anticipation of future developments. The future of biology is impossible to predict with confidence, but certain directions seem likely to continue. Molecular technologies will become cheaper and more powerful, enabling studies at scales currently impractical. Computational approaches will become more important as biological data accumulate faster than can be analysed with existing methods. Interdisciplinary integration, already advanced, will continue as the boundaries between biology, chemistry, physics, engineering, and computer science become less meaningful. Applications in medicine, agriculture, environment, and industry will expand, raising new scientific questions as well as ethical and social issues. Biology provides the foundation for understanding human health and disease. Most major causes of death and disability, from infectious diseases to cancer, to heart disease to mental illness, are fundamentally biological problems. Medical advances depend on biological knowledge, from vaccines that prevent infectious diseases to treatments that target cancer-specific mutations to emerging gene therapies. Personalized medicine tailoring treatment to individual genetic profiles represents one frontier where biological knowledge is being translated into clinical practice. Limits to what biology can achieve in medicine should not be overlooked, but neither should the progress already made and the potential for future advances. Agriculture, which sustains human civilization, is an applied biological science. Crop improvement through traditional breeding has increased yields dramatically over millennia, with modern molecular breeding and genetic engineering accelerating this progress. Pest management relies on understanding pest biology and ecology, including integrated pest management approaches that reduce reliance on chemical pesticides. Soil biology, including the microbiome of agricultural soils, influences fertility and sustainability. Climate change poses challenges for agriculture that will require biological as well as technological solutions. Environmental management applies biological knowledge to conservation, restoration, and sustainable use of natural resources. Understanding ecosystem function guides efforts to maintain services that human well-being depends on. Restoration ecology applies succession and community assembly principles to rebuilding degraded ecosystems. Wildlife management balances species conservation with human interests and uses ecological knowledge to achieve conservation goals. Climate change adaptation will require biological understanding of how species and ecosystems respond to novel conditions. Biotechnology transforms biological knowledge into products and processes. Pharmaceutical biotechnology produces therapeutic proteins, vaccines, and other medicines. Industrial biotechnology uses microorganisms to produce chemicals, fuels, and materials. Agricultural biotechnology has generated crop varieties with improved traits, though these have been controversial in some contexts. Emerging biotechnologies, including synthetic biology and gene editing, promise new applications while raising questions about safety, ethics, and governance. The philosophical implications of biological knowledge are profound, though interpreting them is not straightforward. The evolutionary origin of humans challenges some traditional views of human nature and human uniqueness. The neural basis of mind raises questions about consciousness, free will, and personal identity. The possibility of life elsewhere in the universe has implications for humanity's place in the cosmos. These philosophical questions are not resolved by biological facts alone, but require philosophical analysis informed by those facts. Biology, as a discipline, has grown from natural history observation to experimental science to the current data-intensive, computationally enabled field. This growth has fragmented biology into specialties that sometimes have difficulty communicating with each other, even as it has generated unprecedented understanding. Integrative frameworks, whether evolutionary, ecological, or systems-oriented, attempt to hold the field together and provide common language. The diversity of approaches, subjects, and methods is both a strength, allowing many questions to be addressed, and a challenge, requiring effort to maintain coherence. The living world remains full of mysteries despite centuries of study. Why do organisms age? How does consciousness arise from neural activity? What determines the diversity of life in different places and times? How did life originate? These questions remain open, with current understanding providing at best partial answers. The humility appropriate to these limits does not diminish the achievement of what has been learned, but rather motivates continued inquiry. Biology continues to offer discoveries that surprise, technologies that transform, and understanding that illuminates the nature of life itself.